Cell suspension culture. Cell suspension culture. Cell suspension cultures are rapidly dividing suspensions of cell growth. Cells grown in liquid medium grow more rapidly than callus culture and more ready to culture manipulations. Comprised of cell aggregates and dispersed single cells. Techniques of cell culture. In the initiation of cell suspensions culture, fragment of undifferentiated callus, liquid medium, aeration, subculture, agitation, suspension, cell cultures. Initiation of cell suspensions. Initiation and establishment time depends on plant species and growth medium. Dicots are easier to establish in suspensions than monocots. Initiated by agitating a fragment of callus in a volume of liquid medium on a shaker. Three producers used for cell dispersion. Initiate from friable callus, non-friable callus, callus treated with cell wall degrading enzymes. Initiation from friable callus. Most commonly used starting material, easily fragmented during agitation in liquid medium, achieved by callus passaged on a 7-day cycle for 2 to 3 weeks. 2 to 3 weeks. Ratio of auxin or cytokinins altered, increase auxin concentration. Use 2 to 3 gram of friable callus per 100 ml of liquid medium. Low levels of callus tissue fail to replicate. Subculture the cells to fresh medium at a ratio of 1:1. Filter the actively dividing cell suspensions to remove large callus aggregates to get fine suspensions. Initiation from non-friable callus. First, repeatedly subculture or transfer callus fragments to semi-solid medium until friable. Initiate and establish cell suspensions. Initiation from callus treated with enzymes. Pectinase cellulase breaks down middle lamella of cell wall and separates plant cells. Maintenance of cell suspensions. Variety of culture vessels available. Suitable ones allow large surface area to maximize gas exchange. 20 ml culture medium plus 100 ml flask. 50 to 100 ml in a 250 ml flask. Flasks enclosed with sterilized or dried aluminum foil caps. Aluminum foil caps. Shaker speed 100 to 120 rpm with optimum incubation conditions. Small batch culture volume in a small fixed volume less than 100 ml. Growth characteristics of cell suspensions. Plant cell suspensions consist of cells with diverse morphology and state of aggregation. Two morphological types of cells can be distinguished: cell aggregates made up of small cells, large and elongated single cells. Proportion of the cell type depends on the passage of culture and nature of oxygen. Monitoring growth of culture, cell number, PCV, fresh weight and dry weight, cell viability, medium conductivity and pH. Use to use two methods simultaneously till stationary phase. Growth curve of suspension cultures. Plotted for fresh dry weights and PCV gives the length of culture cycle used to decide the subculture interval of suspensions. Lag phase, exponential phase, linear phase, decelerating phase, stationary phase. Use of cell suspensions study various factors and compounds affecting growth and differentiation. Study cell division, rapid preparation of protoplast, large scale production of commercial plants via somatic embryogenesis, commercial production of secondary metabolites. Anther or microscope culture. Introduction. Anther or pollens can be cultured on a suitable medium containing sucrose, usually 2% iron, vitamins, hormones, etc. The hormonal component of the medium is important for initiation of growth. Usually to the culture medium, auxin, cytokinin, etc. added either singly or in various combinations. Low concentration of auxin stimulates callus formation in a medium supplemented with auxin. Embryoid formation usually occurs at a faster rate as observed in anther culture of datura. Anthers cultured on a medium containing coconut milk or keratin develop embryoids which later form haploid plantlets. Callus is formed from pollen grains on a medium supplemented with yeast extract or season hydrolysate. Factors affecting anther and pollen culture. Activated charcoal. It has stimulatory effect on embryogenesis, and this has been observed in anther culture of potato, rye, tobacco, etc. This may be due to removal of inhibitory substances from agar by activated charcoal. Charcoal may absorb the degradation product 5 to 2 furfural of sucrose. Anther cultures of petunia and nicotiana indicate that activated charcoal removes both exogenous and endogenous growth hormones from culture medium. Temperature has significant effect on pollen embryoid development in datura. Embryoids are not formed if cultures are maintained at 20 degrees centigrade or below. In Nicotiana tabacum, optimal temperature for embryoid growth is 25 degrees centigrade. Of pre-treatment of anther at 3 to 10 degrees centigrade for 2 to 30 days stimulates embryogenesis. Wenzel 77 observed that buds of scale cereal pre-treated pre-treated at 6 degrees centigrade for 6 to 10 days, develop embryos in and become. If the birds are pre-treated at 5 degrees centigrade for 72 hours, then 58% anthers produce embryos. Sometimes pre-treatment at high temperature helps embryoid formation in Brassica campestris. Pre-treatment of anthers at 35 degrees centigrade for 24 hours help embryoid formation. Centrifugation of the anthers at 3 to 5 degrees centigrade for approximately 30 minutes helps embryoid formation. Stage of the anther. Particular stage of the anther at the time of culture is important. Usually anthers just before or immediately after pollen mitosis are more suitable for culture. Suitable stages of anthers for culture are pre-mitotic, mitotic and post-mitotic. Pre-mitotic stage. Anthers at this stage have microspores which have just completed the first meiotic division, and the pollens are immature, uninucleate and starch-free. Anthers of Hodium vulgare and Hyoscyamus at this stage are suitable for culture. According to Nietzsche 72 and Sunderland 71, anthers with uninucleate pollens are suitable for culture. Mitotic stage. In some plants, anthers at first pollen division stage are not most suitable for culture, as and Datura inoxia. Post-mitotic stage, early bicellular stage of pollen development is most suitable for culture in Atropa, Belladonna and Nicotiana species, etc. Mature anthers are usually unsuitable for culture, but in Brassica oleracea, mature anthers are the proper stage for the anther culture. Anthers of proper stage are chosen by selecting flower buds of definite length under fixed environmental conditions. Photoperiod and light intensity. Higher number of embryos are formed when anthers are taken from plant grown under short days and high light intensities. Flowering time. Anthers taken from flowers at the beginning of the flowering period of the plant are more suitable for culture. Endogenous auxin. Embryogenic pollens are found near the tapetum within the anthers. The tapetum may release some substance which initiates embryogenic development in pollens. This is observed in Hyoscyamus niger by Ragavan. Age of the plant. Usually anthers from younger plants are more suitable for culture. Methods of anther culture. Selected plants are cultivated until they reach flower bud stage. In some cases, flower buds are chilled few days prior to culture. Flower buds of proper size and developmental stage are taken and surfaces sterilized with alcohol or hypochlorite solution for 10 to 20 minutes. Buds are rinsed several times in sterile double distilled water. The anthers are carefully excised from flower buds using force-fed dissecting needle. Filaments must be removed prior to culture, otherwise callus may be formed at the cut ends. Anthers may be cultured either on agar solidified culture medium or placed on a filter paper bridge